Nope. Okay, I think we're finally ready to start here. And we've got almost a full house. And okay, I don't need to know that. And we're gonna wrap up, as far as I could tell from checking the video, just to double check myself, we still have to finish up proteins in chapter 2. I thought we were done with chapter 2, but apparently we're not. And this won't take very long this morning. If I'm wrong, please someone correct me, because I know you don't want to hear me say it again, talk over stuff again. But it is Monday and things happen. So, okay. Anybody have any questions on anything? Okay. Then I'm going to go into chapter 2 one more time and get rid of that and finish that today. And then we can start on chapter 3. Yes, what an amazing concept. So, okay, here we go. So we're talking about proteins. Proteins show up in four structures. The first structure, the way the cell makes the protein, doesn't last very long. As soon as it gets made, it comes out in a straight line. When it's being manufactured, the protein gets shaped into it comes out it chugs out of the cell as a straight chain. But it doesn't stay that way because it immediately starts getting twisted and bent into its functional shapes. See, proteins work a lot like a key and a lock. They fit on cells and do things for us, like hormones or enzymes or structures like that are dependent on their shape. They can't just land on a cell and work, just laying on top of the cell. They have to twist themselves around because they have sites on their chain that are going to fit into target areas on the cell. So the first thing that happens is we see that the protein chain gets twisted and it gets bent and it gets wrapped around itself. The secondary structure, there are two of them. As the primary structure gets bent around, its secondary structure forms. And the secondary structure can either be an alpha helix looking like a spring or a pleated sheet that looks like an accordion type of an expandable ribbon, if you will. An alpha helix shows up where we're going to have a lot of elasticity, a lot of rebounding in here. The elastic protein we have in our skin is made up of lots of alpha helix structures. The coils here, they can expand and contract, and come back to our original shape. So the elasticity we get from our skin and in cartilage, things like that, places where we have give and take, we're going to have a lot of alpha helix. The pleated sheets occur where we have lots of bands of protein lined up, one next to another, next to another, next to another, where we want to have great strength. like the tendons that attach bone to muscle. The tendons are made of collagen, which I've said many times, the most abundant protein in the body. But when they line up, they give you an incredible amount of strength. There are just little threads here of protein, but those threads actually have greater strength than a corresponding thread of stainless steel. So when they line up in rows and can stay flexible, we use them for holding joints together, attaching bones to muscles. We use them to give great strength to the underlayers of our skin. So now a protein isn't just going to have alpha helix coils or pleated sheets in there. They're going to have mixed combinations of both, occurring. They form what's known as a tertiary structure, where we have multiple occurrences of these alpha helix and pleated sheets in here. And then we form a quaternary structure, where the protein folds over on itself again and again. And it usually has 4 segments to it. And a lot of the bending and twisting is due to the formation of hydrogen bonds between different sections of the protein strand. So here's our, at the top here, here's our primary structure right there. This is a very poor illustration of our alpha helix, but imagine a spiral staircase, and that's, or a spring, or a telephone cord, on an old phone, on the handset. And the pleated sheet would be, take a piece of paper and fold it over in itself 10 times, and you have this zigzag shape. That's the pleated sheet. Notice how the proteins are lined up in a row, but they also have hydrogen bonding occurring with the various strands within the protein itself. It gives us great strength. The tertiary structure is a combination of alpha helix and pleated sheet. And then here we see a quaternary structure, often described as having multiple tertiaries assembled there. The classic example of a quaternary structure is hemoglobin, the respiratory pigment that our bodies use to carry oxygen. So proteins fall, so we see the 4 basic structures here. Proteins also can either be considered a fibrous protein, like muscle, or a globular protein, like a hormone. Fibrous proteins are the structural proteins. These are, they come in strands, they are insoluble in water, they're very stable. They're usually tertiary or quaternary structures. They give us great strength. The keratin, waterproofing in our skin, the elastic fibers in our skin, the collagen, and the contractile fibers, the actin and the myosin that shows up as protein in our skeletal muscle and smooth muscle and cardiac muscle. These are the fibrous proteins. They are very stable. They're very strong. They can take a lot of punishment. They can survive changes in temperature and pH very easily. They're very they're very important to us. Globular proteins are different. Globular proteins are squishy. They are round. They are soluble in water, and they're very sensitive to changes around them, including temperature and pH. They also have tertiary and quaternary shapes. Hemoglobin is a good example of a quaternary protein. Or all the enzymes are globular protein. Hemoglobin is a globular protein. The enzymes are all globular proteins. The enzymes, these globular proteins, usually have very specific areas that we call active sites. And these active sites are where chemical reactions will take place. Whether this is where a molecule binds to the protein or the protein binds to a target cell. And the antibodies that our bodies produce in response to pathogenic bacteria or funguses or pathogenic viruses that our body responds to the indicators on these pathogens that that they're a foreign substance and we need to attack them, so they make these antibodies. Hormones are mostly protein. There's only a handful of hormones that aren't protein, and those are the steroid-based hormones. We have something called molecular chaperones. They work sort of like a mixer or a blender, because they take a straight chain protein and they twist it around into its secondary and tertiary structures. And then, of course, the enzymes. Enzymes are the biological catalyst. You can see here that we have an enzyme in our mouth called amylase. And, when we eat carbohydrates, like a piece of bread, an apple, a potato, chips, whatever food we eat, the carbohydrates are made of starch. Bread's made of starch. Rolls are made of starch. Biscuits are made of starch. Now we start the process of digesting this starch in our mouth. Sort of a trick question. Where do we start the digestion? Everybody says the stomach. We start it in our mouth. We start with an enzyme known as amylase. Amylase will bind to, amylase is just an enzyme. Amylase is an enzyme that binds to the glucose molecules in starch. Now, starch is made up of thousands and thousands of glucose molecules. Amylase binds to the long chain of starch with thousands of glucose molecules and starts breaking up the starch into sucrose. It breaks them off two at a time. So we get sucrose or maltose or whatever disaccharide we have. And then the disaccharide is going to be further digested later on. So we start the process of breaking down a starch in the mouth. Now, amylase has what are known as active sites located on it. The active sites are designed to fit on starch and only on starch. Nothing else will fit there on these active sites. And these are the functional regions of the enzyme. The active site is looking for what are called substrates that fit there. And so the amylase breaks down the starch into maltose, which is a disaccharide very similar to sucrose, table sugar. The problem with the globular proteins is they are very sensitive to changes in temperature and environment. The fibrous proteins are very stable. They don't respond to change like that. But these globular proteins can become denatured. A change in pH, a change in temperature can cause the active site of the enzyme to become altered. And so the substrate, the agent that you're trying to get to land on the enzyme can't land there anymore because the site for it to land on is destroyed. Now, sometimes they get warm, they change their shape a little bit, and then the temperature goes back down to normal, and the shape resumes, goes back to its normal shape. Sometimes, many of these active sites can be mildly denatured and then reverse back to their normal shape if the swing in temperature or pH is moderate. Extreme changes are not reversible. Extreme changes in temperature are not reversible. Consider this. Look at the egg there. Now, the white of the egg is made of the protein albumin. So it's albumin's a protein we have in our own bodies in our plasma. But anyway, albumin, you crack open an egg, you got the yolk, and you got this reasonably cleared gel that comes out containing the yolk, and that's the albumin, or what we call the white of the egg. That's all protein. When you cook that albumin turns white, it becomes denatured, and you can't reverse that process. You can't take the denatured albumin and convert it back to the transparent gel. Once it turns white, once it's cooked and totally denatured, all you can do is eat it. There's nothing else you can do to it. These changes, these denaturation processes, these becoming denatured, is not a reversible process. And we talk a lot about enzymes right now because enzymes are very important to us. Enzymes, anything that ends in ASE, like amylase, catalase, hydroxylase, carboxylase, any of these are going to be enzymes. Enzymes always end in the ASE enzyme. They are what we call the biological catalyst. Remember, in chemical reactions in our bodies, we can't change the temperature. We can't change the particle size. We can't change the concentration because we're already dealing with very small particles to begin with and very small concentrations. All we have, and we certainly can't do anything about the temperature. All we can do is use a catalyst to make the reaction go faster. And so while we're dealing with small concentrations, we can flood an area with millions of enzymes. And those enzymes will go out and grab the reactants and bring them together. That's all an enzyme does. It brings reactants together. See, reactants are going to be bouncing around anyway. Reactants have a certain amount of kinetic and potential energy. Everything does. So the reactants in our bodies are bouncing around, and if they randomly run into each other, a chemical reaction occurs. But that's pretty slow. We can't wait for things like that to happen. So what the enzyme does is you fill up the spaces with enzymes, and those reactants are grabbed by the enzymes. Like there's a million enzymes in the space there, and they're competing to find these reactants. Reactants are grabbed by the enzymes, brought together, and a chemical reaction ensues. So now we call the reactants the substrate. We call where they land on the enzyme the active site. So what an enzyme does is simply grabs these substrates, brings them together, a reaction occurs, and a product is made and then released. And the enzyme can go do it again and again. The enzyme doesn't get changed or altered by this reaction. So instead of having a handful of reactions per minute, we can get millions of reactions per minute. So it's our workaround around the limitations of temperature and particle size and concentration. In other words, it lowers the amount of energy necessary for a chemical reaction to occur. So you can see there's a substrate, they come together, they're brought together on the enzyme at the active site. Bringing them physically together causes a reaction to occur, and we release the product. Now, these enzymes, these globular proteins, consist of a protein portion and an area called the coenzyme. Coenzymes are not enzymes. They are not, wannabes. They can't be converted into an enzyme. The cofactor or coenzyme are just part of the structure. They make the overall enzyme work. The protein depends on either a metal ion or a molecule from a vitamin. Vitamins are coenzymes. Vitamins make these chemical reactions go faster. That's why we take vitamins. The vitamins are very specific for what they can do. The enzymes are very, very specific. Amylase, for example, only works on starch. It doesn't do a thing on any other structure. It doesn't allow amino acids to come together. It doesn't break down fats. It just breaks down starch. That's all it does. Sometimes the reaction is a breakdown reaction. Sometimes it's a build-up reaction. So the enzymes are very, very specific. So we don't, the substrates, are, as we say, the enzymes are substrate specific. The reactants only, it's like a lock and a key. The key will only work in certain locks. And if the substrate is the key, it only fits in certain places. And this is an important thing to remember because, and I will tell you that no one has ever come up to me and asked me about activation energy, but we know that enzymes work because they lower the activation energy necessary for a reaction to occur. It takes less energy for chemical reactions to occur in our bodies when we use our enzymes. Without enzymes, these reactions don't occur. Instead of depending on the reactants just to bounce into each other and randomly come together, we can make millions of reactions occur at once. We don't have to spend all that energy, bounce, bounce, bounce to get things to happen. Oops, let's see if this is going to work for us today.
An enzyme is a protein that changes the rate of chemical reaction.
That was a hard-hitting little factoid there. It took more time to set it up than for it to play. But what it does is, if this is an enzyme, this is the active site here, and the reactants, the substrates, will be drawn into that active site as they bounce in there, because there's going to be millions of enzymes where the reaction should be taking place, and so those reactants, the substrates, are going to bounce in there, and once they land on the active site, they're going to stay there until the other part of the active site is filled up, and then the reaction is going to occur. And this is a simple little, this is non-animated, this probably works better. There's your enzyme, there's the active site, there are the substrates. This is the assembly of amino acids to make a protein. And we have two amino acids that are specific for this active site. We call, these are the substrates. They land on the active site. They're the only ones that will fit there. And a peptide bond forms, and we release the amino acids now linked together with their peptide bond, and the enzyme goes off to go do it again and again and again. And then another enzyme will link together, make another peptide bond to link together even more amino acids. So, now when the enzymes denature, the active site gets damaged. See, what you're seeing here is with a normal healthy enzyme, the substrate lands on the site perfectly and it fits. But when the enzyme is denatured, the active site is altered and the substrates can't land there. They don't fit anymore. Now, if it's a mild event, like a slight rise, a moderate rise in temperature, maybe 104, 105, or thereabouts, maybe even as high as 106 or 7, or a decrease in temperature, there's going to be the active site can be mildly denatured. If it continues and goes up even further, or if the pH drops or rises, then the active site's going to be permanently denatured and can't get reversed. This is something we pay very close attention to when we have extremes in body temperature and extremes in pH. Because if we start denaturing, said it right that time, we start denaturing the active sites, the enzymes cease to function and chemical reactions are going to start slowing way down. Yeah. So that leads us into the last segment here, the nucleic acids. The nucleic acids are made of the same molecules, atoms, if you will, found in the other types of organic molecules. You know, we've looked at carbohydrates, we've looked at lipids, we've looked at proteins. The nucleic acids are the DNAs and the RNAs. They're made of carbon and hydrogen and oxygen and nitrogen because they have nitrogen bases in here. They have phosphorus in here. They are the largest molecules in the body. A DNA strand, a molecule of DNA can be 6 feet long, one giant strand of molecule. These nucleic acids are polymers, you know, of course, long strands, large molecules. They are made up of monomers called nucleotides. The nucleotides have a base, as we say, nitrogenous base or a nitrogen base with a sugar, usually a 5-carbon sugar of some sort, a ribose sugar, and a phosphate. So we have a nitrogen base, a pentose, 5-carbon sugar, and a phosphate attached here. Now, the two types of nucleic acids are DNA and RNA. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. The RNA has an extra oxygen in its sugar, and that's the only difference. The nucleotides are going to be the phosphate group attached to a pentose sugar, a 5-carbon sugar, with a nitrogenous base there. There are 5 different bases. We have cysteine, thymine, uracil, adenine, and guanine. Those are the 5 different bases. And these bases are complementary. So they will bind, you know, they will have complementary bonding occurring. I'll explain that here in a second. So these are the 5 bases: cysteine, thymine, uracil, adenine, and guanine. They are pyrimidines and they are purines. The purines have 2 rings. The pyrimidines have 1 ring. Important factoid, no, not really. But both sets of nucleotides are made of these monomers. I'm sorry, both sets of nucleic acids are made up of these nucleotide monomers. They both have the same structure: a phosphate, a sugar, and a base. The only difference is that the DNA is missing an oxygen off of its sugar. That's why we call it deoxyribonucleic acid. DNA is very important. DNA is the basis of our genetic information. DNA is the instruction manual on how to make us. DNA contains all of our genetic information on how to make all of our proteins. And that's what we are all about. We are all about our proteins. DNA is found in every cell except red blood cells because we've kicked out the nucleus. We don't have a nucleus in red blood cells. DNA is contained in the nucleus of all other cells. It is a double helix. It has 2 strands. side by side, linked together by the hydrogen bonds between their bases. The nucleotides here have a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The purines, the double ring structure, will bind to the pyrimidines, the single ring structure. We have in DNA, and this is what's important to remember, is we have adenine, guanine, cytosine, and thymine. Adenine, guanine, cytosine, and thymine. The adenine will always bind with thymine, always. It's supposed to always bind with thymine. Now, the error rate in a DNA strand when it's manufactured for us is about 1 in a billion. That's pretty good. And we have and we get into chapter 3 here shortly. We will eventually discuss the mechanisms that DNA has to repair itself so that it can fix mistakes that may occur. So but adenine will always pair with thymine, and guanine will always pair with cytosine, always, unless there's a mistake. But the mistake rate, as I said, is about 1 in a billion. And that's usually before we fix it. So the nitrogen bases in DNA, adenine, guanine, cytosine, and thymine. Adenine pairs with thymine, guanine will pair with cytosine. This is what a DNA molecule looks like. It's a double strand. The bases are attracted to each other on the strand. As the strand is laid down on both sides, the bases develop hydrogen bonding across the strand. These are hydrogen bonds right here in the middle. And here we see there's guanine bound to cytosine. There is thymine bound to adenine. There's adenine bound to thymine. And here is cytosine bound to guanine. So it doesn't matter which side they're on. Adenine will bind to thymine. Thymine will bind to adenine. Cytosine binds to guanine. Guanine binds to cytosine. And they're held together. The two strands of the DNA molecule, it's a double strand, are held together by hydrogen bonds that occur between the bases. So when we say adenine binds to thymine, it's because a series of hydrogen bonds occur from the adenine to the thymine and the thymine to the adenine. This is the complementary base pairing. When we say adenine binds to thymine and only thymine, that's complementary base pairing. The bases pair up, adenine to thymine, cytosine to guanine. Hydrogen bonds form there, and the shape of the molecule is determined by the hydrogen bonding. The hydrogen bonding causes the molecule to twist, and that forms the spiral, the circular spiral shape, the spiral staircase shape, or the twisted ladder shape. Imagine a very long ladder that was very flexible that you could twist it, and it would twist into a spiral. That's what we see here. And it is the rungs of the ladder, these structures here, that form from the hydrogen bonds. The hydrogen bonds twist the molecule. Now, RNA is similar to DNA. It's also made of a nucleotide with a phosphate, a sugar, and a base. But the sugar is not deoxyribose, it's ribose. It's a single sugar. It's a 5-carbon sugar. But RNA doesn't form a double strand. So first of all, with RNA, it's a ribose sugar. And second of all, we don't contain thymine in the RNA. We contain uracil. We substitute uracil in here for the thymine. And so RNA as a single strand becomes the workhorse. The RNA leaves the nucleus. The DNA never leaves our nucleus. The RNA goes out into the cytoplasm and controls the assembly of proteins. There are three versions here of RNA. We have messenger RNA, which does exactly what it sounds like. It takes the information from the nucleus out to the cytoplasm. We have transfer RNA, which brings amino acids together to be assembled. And we have ribosomal RNA, this is used to make a structure called the ribosome, where the protein assembly takes place. DNA doesn't do a thing except contain the instructions and direct the RNA to go do something. The RNA does all the work under the direction of the DNA. The DNA never leaves the nucleus, so the DNA never gets altered by the production of any kind of protein. Okay, the order of the bases. The bases can be in any order, but the sequence of the bases are going to be organized in clusters of threes called triplets. And each triplet is specific for an amino acid. The DNA is our code, our cookbook on how to make proteins. It's our instruction manual on how we function. Everything we make is based on protein. Everything our structure is based on protein, and the ability to replicate and repair and rebuild and replace everything is based on protein. The instructions on how to do this are based on the sequence of the bases in this triplet code. Now, a gene is nothing more than the physical location on the DNA strand for information on a specific trait. How to make bone, how to, what is our blood type? Can we, do we have this is a true fact, do our earlobes dangle or are they attached to the side of our head? Do we have freckles? Do we have dimples? Those are all powerful genetic traits. Can you curl your tongue or not? That's a genetic trait. But other things are more important: body stature, hair color, eye color, skin color. How our physical appearance is all based on the structure of the genes in the DNA. All the information contained in the DNA is clustered into these genes, which is nothing more than the physical location, location of these bases, the nitrogenous bases arrayed in triplet form, clusters of 3 at a time. Of the various bases, adenine, and you only have 4: adenine, thymine, guanine, and cytosine in DNA. The order of those bases determine the amino acid that's going to be assembled in the protein. Proteins are made up of amino acids. Proteins are made of amino acids in a specific pattern. What is the sequence? What protein goes here? What amino acid goes here? What amino acid goes next? What's the one after that? And while there are only 20 amino acids, as I said the other day, you can, in a dictionary, you've got about 350,000 words made up of 26 letters in the alphabet. So 20 amino acids gives you plenty of flexibility here to make proteins of any length you want. So all the proteins in our bodies are dictated by the DNA. The DNA contains the order of how to assemble these proteins. So the instructions are all there in the DNA. And we'll talk a lot more about that in chapter 3 here shortly. Okay, the last part, the third nucleotide structure is based off of an RNA nucleotide, and it's called adenosine triphosphate. And this is what we use for currency in our cells. We know we get our energy from carbohydrates. We break the carbohydrates down to glucose. Then we oxidize, we burn the glucose. When we oxidize a molecule, we break it apart. We release the potential energy from the electrons. We break the covalent bonds holding that molecule together, and we release potential energy. That's what we do when we burn something, when we oxidize something. We strip off the electrons, and we release energy. Now, that energy has to go somewhere. And we transfer that, when we oxidize glucose, we park that energy in ATP. Because ATP is what we use to move that energy around our bodies. We just can't have raw energy from glucose bouncing around. We have to put it somewhere. So we store it in the ATP molecule. The ATP molecule has, a sugar, a phosphate, and a base. The base is adenine, and it's a 5-carbon sugar, and it has not 1 phosphate, but 3 phosphates attached to it. That's why we call it adenosine triphosphate. The molecule looks like this. The 3rd phosphate in adenosine triphosphate only attaches to the rest of the molecule through what we call a high energy bond. It's a very fragile bond. when we have available energy, like we've oxidized glucose, we can take inorganic phosphate and we can take what's known as adenosine diphosphate, that's ADP, bring them together. And now we have ATP, and we can now ship that ATP around our bodies anywhere where we need energy for activity, like muscle contractions. We need ATP, we need energy for muscle contractions. So we send them ATP. We break the high energy bond and release the energy into the cell, into the muscle cell. And we end up with phosphate, and we have ADP. Now they're not destroyed in the reaction. They just break apart into their separate components. And then when we get more energy, we put them back together again. So we're always going from ATP to ADP, and then ADP plus a phosphate, and there's always phosphates to form ATP again. The bad news is that ATP only lasts for 7 seconds. So we have to constantly be making it again and again and again. And as long as we have glucose for energy, we will continue to make ATP to power our bodies. Okay, and that's the end of, finally, chapter 2. Let me get out of here for a second and go into chapter 3. There we go. So this is the start of a brand new unit. The start of a brand new unit. Test 1 covers chapter 1, the introduction, and chapter 2, chemistry. This is going to be part of this next section. Chapter 3 is going to be part of test number 2. So that's quite a ways off. So, okay. Looking at cells, our body is made up of billions of cells, 200 different types of cells in the human body. Now, the concept of the cell and the cells were living structures didn't develop until the 1850s or somewhere thereabouts, when the cell theory was finally proposed. The cell theory has 3 parts to it. Every living structure is made up of cells. Every living structure is made of cells. It seems rather simplistic now, we know that, but in the 1850s, nobody was, weren't certain yet. Cells are the basic living unit of structure and function in organisms. In other words, cells are the smallest living unit around. There's nothing smaller than a cell that is alive. And third, living cells only come from other living cells. There is no artificial creation of life. Living cells only come from other living cells. So that's the cell theory. There's no proto-life that you can add to get mixed together and create a true living cell. Can't do it. Now in the body, as I said, we have over 200 different types of human cells with a wide range of diversity in structure and in function. Some cells are very long. Some nerve cells are over 4 feet in length, like the sciatic nerve is made up of thousands and thousands of individual cells linked together to make this nerve, and they run from our hip to our great toe, so it can be 3 or 4 feet in length. Cells can be squishy, like red blood cells and white cells. for carrying oxygen and for fighting infection. We have muscle cells that can contract. We have nerve cells that generate electrical impulses. All these cells are going to be different. They all do different things. Some absorb nutrients, some filter out waste products, some stretch, some don't. Yeah. Some cells are trapped in a matrix and others aren't. Others are free floating. So cells are, there are 200 different types, over 200 different types of cells in the body, and they all have different structures and different functions, but they're all the same in that they all have three basic components. They all have a cell membrane, they all have a nucleus, and they all have a cytoplasm, except red cells. Red cells don't have a nucleus. They're sort of the exception. So cells, living cells in the human body, all have an outer boundary: the plasma membrane, the cell membrane, the semi-permeable membrane. When you looked at those slides of squamous cells from the prepared cheek slides, you could see, even if we had done it on our own cells, you would not have seen the membrane, but you would have seen the edge of the cell. The cytoplasm, or cytosol, the newer term now, the cytosol is the fluid component or gel-like substance inside the cell membrane, and then within the cytoplasm is going to be a nucleus where the DNA is located. Every cell except red cells have a nucleus with their DNA. Now, even the red cell had a nucleus at one point, but it kicks out its nucleus during the development of the red cell, almost to the end of the process. It kicks out its nucleus so that it has more room to carry hemoglobin. So there's your typical cell. Not every cell is going to look like this, but every cell will have a cell membrane, the plasma membrane, the semi-permeable membrane, the phospholipid bilayer membrane, a lot of names for it. The whole purpose of the cell membrane is to keep the outside out and the inside in. The fluid inside the cell, the intracellular fluid, is pretty much identical to the extracellular fluid, the interstitial fluid that leaks out of the plasma. We don't want to have it running across, flooding back and forth across the membrane, so we have a barrier. So the inside is different from the outside. The cell can control what goes in and what comes out. It's very, very selective. And so we say it is semi-permeable. It is a phospholipid bilayer. It is selective. It's restrictive, whatever. Because first of all, we know it's water hating because it's made of phospholipids, which are fats. And so water is not soluble in fat. So things that are water soluble can't slide through the membrane very easily. Only things that are fat soluble can slide through. So, and of course we see our big nucleus right here in the middle. Now the cell membrane consists of phospholipids, where we take a regular triglyceride with its 3-carbon glycerol and its 3 fatty acid chains, and we knock off one of the fatty acid chains and replace it with a phosphate. So we still have a phosphate head and 2 fatty acid tails. And this is the phospholipid structure that makes up the bilayer. Now, the membrane consists of a double layer of these phospholipids. That's why we call it a bilayer. The phosphate heads are arrayed on the outside of the cell membrane and on the inside facing the interior of the cell, and with the fatty acid tails in the middle. And so this is the lipid bilayer. On the surface of the cell membrane are proteins that are floating all over the place. We call this structure the fluid mosaic pattern. The surface of the cell membrane is very, very busy. Imagine if you had a water balloon that you soaked it, rolled it in, you dunked it in olive oil. So it's very slippery in here. And then you rolled it in a bowl of Lucky Charms. So the reason I picked Lucky Charms is because the cereal and the marshmallow bits have different shapes. They represent different structures. So now they're stuck to that oily coat on the water balloon. The water balloon is squishy. Cell membranes are squishy. They're slippery. The Lucky Charms now floats across the surface of the water balloon. The water balloon, is squishy, and as it moves around and squishes up and down, the cereals and the marshmallows slide across the surface. That's what happens to a cell. This fluid mosaic model means that these raised structures, these portions of the molecule of the membrane, are constantly in motion. And an important one of these structures that form is called the Glycocalyx. The Glycocalyx is the cell identifier. It identifies it as self. It identifies it as us, so that we know that that's one of our cells. Every cell in our body has a glycocalyx structure or structures that identify it as me or you. And as long as we have that, we're fine. If we don't have that on a cell, it's identified as foreign. Bacteria, viruses. Viruses are not living, by the way. They're just a strand of DNA surrounded by a coat of protein, but it has glycocalyx structures also. And it's identified as foreign. Funguses, any kind of pathogen that enters our body is going to have a foreign glycocalyx structure. Transplanted tissue or transfused blood of the wrong type will have the wrong glycocalyx structure, and that will trigger our immune system. And sometimes when we have an autoimmune disorder, our own immune system attacks a cell, even though it has the right glycocalyx structure, but it sees it as foreign. Our cells are also held together by what we call cell junctions. There's 3 different types of cell junctions. We'll talk about that in a little bit here. But our cells are held together with very sophisticated structures to give us great strength, but also great flexibility and great communication. So this is what our phospholipid bilayer looks like. There's the phosphate heads here, and there's the phosphate heads there, and in between are the tails. The phospholipid bilayer is we say it is selectively permeable because it only lets in the things that it wants to let in. It will only let in things that are fat soluble and very tiny, like carbon dioxide can leave and oxygen can come in. Water can't come in, glucose can't come in, and sodium can't come in unless they have a lot of help. And they get that help because we've got to have glucose. We see the transfer of water back and forth, and we see the transfer of sodium into the cell and out of the cell. So we have to have help for some things to get in. Now there's your phospholipid. A little more detailed diagram than what we saw back in chapter 2. It is the phosphate there's a phosphate that substitutes for the third fatty acid chain. And so that gives us a phosphate head at one end of the molecule, and we have two fatty acids left over as the tails. One of the fatty acid chains will have a double bond, so there's a kink there, it's unsaturated, and so now we don't get solid, we stay liquid in the area in between the membrane, in between the outer layer and the inner layer. So this is the tails are what we say hydrophobic, water hating. They reject water, they repel water. It's like oil and water, or you have an oil and vinegar salad dressing. You shake it up, but then eventually the oil comes to the top, and the vinegar, which is mostly water, is below that. And so it's immiscible. They don't mix together. So this is the same thing here in the cell membrane. Water soluble structures or molecules can't get through. And again, this is how the bilayer is set up. Bilayer, because it's two layers. The hydrophobic tails, the water-hating tails. We have the phosphate heads on the inside of the cell membrane and on the outside of the cell membrane. So it's a double layer here. And it looks like this. This is a pretty good representation of what the cell membrane looks like. It's a very busy location. A lot of activity going on here. And it's moving up and down all the time. It's squishy. We have phosphate heads here. We have phosphate heads underneath. We have proteins that stick up that have fats attached to them. We call them, or rather we have sugars and fats together. Down here, here's a glycolipid. Think of a funnel cake. It's a fat and a sugar, a lipid with a carbohydrate. Down here is a sugar-coated protein, a glycoprotein, if you will. It's sticking up here. This could be a glycocalyx. That's what a glycocalyx is. It's a sugar-coated protein. This is a protein that goes all the way through the membrane. It's called an integral protein. This is a peripheral protein. That could be an enzyme. It can be a reactive site. It can be something. Here we see this is cholesterol. Cholesterol, the four-sided ring structure here, is embedded in the cell membrane like rebar in concrete for reinforcing and strength. This is a channel protein. This could be a carrier protein in here. We have two different types of proteins that will go through the membrane to allow the passage of substances that we need inside the cell or we want to get rid of outside the cell. So a very busy surface, and it's always in motion. It's always in motion, always moving around here. It is the undulating surface, if you will, like walking across the top of a trampoline. It's moving up and down, and things are sliding across it at the same time. The bilayer is mostly phospholipids. It has a little bit of glycolipids thrown in there. Those are the sugar-coated fats. And then the rest of it is cholesterol. Cholesterol is very important in our cell membrane. It gives it strength. It gives our cell membrane lots of strength and stability. Now the proteins that go through the membrane can be an integral protein, going all the way through. That's what this purple thing is here. That's an integral protein that goes through the membrane. And see, and while it's not a great animation, this shows you the movement of the membrane. The membrane's constantly moving. The heads and tails are always in motion. We never compromise the integrity of the membrane. We don't rip holes in the membrane or anything like that. But the membrane is constantly moving. Now, the proteins that are embedded in the membrane, if they are an integral protein, they will go all the way through. And they are usually going to be a carrier protein or a channel protein. Carrier proteins and channel proteins allow the passage of materials through the membrane into the cell or out of the cell. Peripheral proteins are usually going to be showing up on one surface of the membrane, like an enzyme tethered there to do something. So integral proteins, they are going to stretch across the membrane. They're transmembrane proteins. They carrier proteins, channel proteins. They may be enzymes. They may be receptor sites for hormones, for example. The peripheral proteins, they are generally attached to the integral proteins. They may be on the surface. They may be on the undersurface here. They are enzymes. They are motor proteins that allow for the movement of the cell when the muscle cells contract, for example. It's the peripheral proteins that are doing this. They're used during mitosis and cell division to move the cytoplasm apart. We see them also, the peripheral proteins are for connections. The cells communicate to each other. The cells are attached to each other. They just don't line up next to each other. They're also, some are woven together. Some are essentially bolted together. Some have holes in their membrane, so they join side by side through these openings, so they can communicate rapidly through these little tunnels that go into each cell. The cell membrane proteins can function as enzymes and for transport, receptors for hormones. Many the protein hormones are too big to get into the cells, so we have to do something called signal transduction. We take the signal from the hormone, the protein hormone, like epinephrine, is a protein hormone. It's too big to get in the cell. It's also not fat soluble, but it can communicate across the membrane and send its instructions into the cell. That's signal transduction. Cells join together, the cells recognize each other, and they hold themselves together. Let's take a look at some of these here. The transport proteins can go, we normally think of them as just opening up channels or passageways in the membrane for water to get through, or ions to get through, or to go in and out of the cell. Maybe let glucose in, for example. But we also use them to carry hemoglobin. Use hemoglobin to carry oxygen. We also use hemoglobin to carry some of our carbon dioxide. So those are the transport proteins. Let me go over here to, okay. Most of our hormones are proteins, and the only way these hormones can tell the cells what to do are going to we have to use this signal transduction where we generate a new signal in the membrane to tell the cell, hey, here's what you're going to do now. If you consider how strong a tendon is, tendon is made of collagen, and collagen fibers are so strong that they will snap bone. A tendon will snap bone before the tendon itself tears. The exception, of course, is the Achilles tendon. We'll talk about that later on, but probably because it probably tears more easily because it gets more abuse than any other tendon in our bodies. But tendons will usually snap bone before the tendon will tear. They're that strong. Of course, here are enzymes. We know I just sort of beat enzymes into the ground, but we depend on the myriad number of enzymes found in our bodies for chemical reactions to occur. Now, so those are four examples of what our proteins are doing for us here. The glycocalyx, again, I want to emphasize this enough. The glycocalyx structure is so important. It identifies the cell as self. We don't attack ourselves unless we have an autoimmune disorder. An autoimmune disorder occurs when our own immune system does not recognize our glycocalyx structures on a particular type of cell and attacks it. Maybe it's blood. Maybe it is the layers that insulate our nerves. But we attack. Certainly, maybe it is the beta cells in the pancreas that produce insulin. Maybe they're attacked by our own immune system. The glycocalyx structure, the carbohydrate, the sugar, and the protein attached to it are the identifiers. They have unique shapes. They're like the flag that says, hey, it's us. Don't attack us. They are the markers that allow us to recognize the cells as belonging to us. So the glycocalyx, every cell has glycocalyx structures that we call them antigens. These glycocalyx structures, these antigens, are the identifiers that tell us what's going on. Foreign substances, bacteria, viruses, remember viruses aren't alive, funguses, transplanted tissue, transfused blood of the wrong type are not recognized. And so they're attacked. If you give a patient the wrong kind of blood, then your immune system is going to attack that blood. And it's not going to bode well for your patient. Your patient's not going to like that. They may not survive a transfusion. About three people a year in the United States die from transfusion mismatches when nobody should die. Because if you've ever been in the hospital, you know that before they put anything in the IV line, and they have to check the labels on the bag and on the line and on your wrist. A little thing, little tag, a little thing that you wear around your wrist when you're in the hospital. And yet, it still happens. And about 3 people a year die. These are the glycocalyx structures. They're very, very tiny. this isn't, this is just, these are the microvilli. These are tiny folds in the surface of your membrane. So they're extraordinarily tiny, but they're very, very important. So, cancer cells change their glycocalyx quickly. You would think that we would recognize cancer cells as foreign and attack them and destroy them before they can harm us. But it turns out our immune system takes about 14 days to respond, to start producing enough antibodies to attack a foreign antigen. Well, what happens is that cancer cells change their glycocalyx so quickly that our immune system never gets a chance to identify the glycocalyx as, it can identify it as foreign, but it can't produce antibodies quickly enough. It takes 10 days to 2 weeks for our bodies to make antibodies to any kind of antigen, any kind of foreign antigen, a foreign glycocalyx. The cancer cell changes their antigens faster than that. So we're always playing catch up. We can never seem to get antibodies made to respond to the cancer cells. So these cancer cells, and cancer is defined as simply as uncontrolled cell growth. That's all it is. That's all cancer is, uncontrolled cell growth. And the mutation that caused a cell to become cancerous is not destroyed because we can't. It replicates itself, it changes its antigens so quickly we can't respond. So, last point, and we'll call it a day here. Cell junctions. How are cells held together? Well, not all cells are held together. White cells are separate. Red cells are separate. Flagella, sperm cells with flagella and egg cells that float free. The gametes are free cells. Most other cells are held together. Simple columnar cells are held together to line our small intestine. Muscle cells are held together so we can have contractile action. Now, cells are held together using cell junctions, and the three types are what we call tight junctions, desmosomes, and gap junctions. With tight junctions, we have integral proteins that go across cell membranes. So tight junctions don't allow anything to come through. The membranes are held together, and this is where the cell membranes are fused together. We see tight junctions in our stomach, so the contents of the stomach acids, and the digestive enzymes can't leak out of the stomach. So the tight junctions, the membranes are, it's almost like they're stitched together. No way, there's nothing that can leak out. Desmosomes look like rivets, look like bolts have gone through. They look sort of like a zipper. And they have proteins that have gone across from one cell membrane to another and form this rivet or this zipper-like shape, so that these proteins are mostly keratin. And what happens here is with the desmosome, you have great flexibility, but great strength. Our heart, for example, cardiac muscle cells are held together with desmosomes because we have great flexibility for the contractions every 7/10 of a second, but we have great strength so we don't tear the muscle cells. And of course, gap junctions are proteins that connect and form tunnels that allow the passage of ions and sugars and communication, electrical signals. Nerve cells are held together with gap junctions so that we can send signals through here quickly without spending a lot of time working our way down. We can send them directly into the cells. Smooth muscle cells have these. Cardiac muscle cells have these gap junctions. Okay, that's a good place to stop. We will, let me get out of here. comments. Okay. Oh, did I stop sharing? Oh, tell me I didn't do that. Okay, sorry. I probably forgot to share. No, it's okay. Well, I guess I'll just have to. Thank you for telling me that. Okay. Well, it was only about. It was only a little bit. I can just start this again on Wednesday. We didn't miss a whole lot, so it won't hurt to go over it again. So, okay. I apologize for that. I'm going to get us out of here, and I'll post this video on YouTube later today. So.